you hear me? Yes? Can you hear me? Okay, good. Now we will be starting a new block which is MSK. You already started, but the embryology is concerned is today is the first one. And it will be interesting if you get the concept clear and you realize that you can answer all my questions if the concept is very clear. And we start off obviously with this photograph, which you have seen it 1,000 times. This is 1001. Again, I am showing it because this is the fundamental concept where you are looking at the embryo very early period. If I see this thing, I will say between third and fourth week, maybe 3 layers are there. And the picture is like this way, it's not like this way, it's anterior posterior. You see 3 lines, and 3 lines are made for ectoderm. You know what the ectoderm is doing. It produces neural tube and the neural crest. Often it's called the neuroectoderm, so it also produces skin. Skin is the largest organ we have, and we will be doing it this block, the final block, for the year 1. So ectoderm is concerned, you know that it is forming the skin, neural tube, which is the brain and the spinal cord, neural crest, there are plenty of cells coming from the neural crest. It's the final cells which go to migratory cells, which go to a system to make it functional. Then comes a mesoderm, which has 3 parts, paraxial mesoderm. This is the place where I left it earlier for a reason that the MSK block is entirely on this one, paraxial mesoderm, that we'll be talking. Intermediate, you have done it already, and lateral mesoderm, once in a while I emphasize that it folds actually. This is the somatic one, this is the splanchnic one, and it folds like the next photograph is folding. When you understand, this is the somatic, this is splanchnic mesoderm. But I'll be concentrating on over here, and then you know the endoderm, what is this, all about it, forms a tube for the GI tract and then lung tissue. This is just an overview of understanding the 3 layers which are formed very early period at the outset of the embryonic development. Again, I'm saying that we'll be concentrating on this one, and I try to put a little bit ball-shaped, like a round-shaped structure, often we call somite, S-O-M-I-T-E, somite. Somites are the places like a ball-shaped structure, which has 3 different parts. Somites are present inside the paraxial mesoderm. It has 3 distinct parts. One is called sclerotome, which forms the vertebrae, myotome, which forms the muscles, and the dermatome, you have heard the name dermatome, which is going towards the skin and produces some kind of sensation. That's called the somite. Somites, when we are born, before we are born, we have a very early period of time, we have around 40 to 45 somites. But we do not need that many vertebrae. So we reduce to around 30, 31 vertebrae we have. Okay? So over the time, that is joined together to form one. Now this somite will join with somite, so it makes one vertebra. And this is the neural tube which goes inside. That's where the spinal cord. So now you can correlate how the vertebrae is formed. Vertebrae are formed between the 2 somites joined together, taking the neural tube inside. I'll be repeating it 10 times so that till it is clear. For the time being, I'm saying the somites are the ball-shaped structure which are present in the paraxial mesoderm, and it has 3 parts. One is called the sclerotome. Sclero means it is the bone type of structure. So you have the sclerotome, like you have the vertebrae and the ribs. They all come from the sclerotome. Myotome means, listen to me very carefully. Myotomes are the structures which form the muscles. Which kind of muscles? Skeletal muscle. All our muscles that you know, that you'll be knowing it upper limb, lower limb, all come from myotome. They migrate from here to if this is the arm, so it will migrate to form the arm and the forearm, or the thigh and the leg. Okay, so all these skeletal muscles, if I ask you, you have heard the name now supraspinatus, right? Have you heard the name supraspinatus muscle? Which muscle do you have? Pectoral is major muscle. Have you heard the name?
Yes.
You have heard the name. So if I ask you, it's coming from where? Myotome. Any muscle, skeletal muscle you know now, they are all coming from the myotome. Any muscle. Okay? Any vertebrae is the sclerotome, and the dermatome. We'll talk about the next class, the detailing of the dermatome. You have heard the name probably, dermatome. You have heard the name, but you don't know exactly what this is all about. It's come from the somae. So let me put it together, very holistic view of the limb development. Then I'll go to the slides one by one. Okay, you'll enjoy the slides more when I explain to you a little bit. Not too much, but a little bit. See what's happening. If I ask you the limb development, which week it will start? Fourth week. It's not a rocket science now. Everybody knows by fourth week, the limb development will start. It starts at the two places. One in the cervical area to form the upper limb, one in the lumbar area to form the lower limb. So it starts with a little bit of bud. You know the flower. Flower doesn't come all of a sudden. First is the bud, followed by the flower. Right? Similarly, we have the bud, then it will be forming like a paddle. You see the bolt paddle, and then it will be like a plate. So little bit of initially bud, paddle, and the plate. And then there will be a rotation of those structures. The upper limb rotates laterally. I'm rotating laterally so that the elbow comes posteriorly. Right now, if you see in my hand, the elbow is over there. So in the embryonic life, the baby is like this to start with. Then it rotates upper limb towards the lateral side, like this way. So now the elbow is posteriorly placed. The leg, just the opposite. It rotates medially so that the knee comes anteriorly. This is the final outcome of the baby. In other words, what I'm trying to say, everything starts from the somae. Because for somae to go to a structure, like let's do the upper limb. I have to make the upper limb. How do I make the upper limb? We have somae. We have 40, 43, 45 somae. They will join together to form 30 to 31 vertebrae we have. Okay? So 31 places, what is happening is we have the vertebrae, that's okay. But we have also the muscles, the myotome. So myotome now goes towards the lateral plate, lateral mesoderm, where we have the muscles now coming from the myotome to over here, but we need the bone. So bone and the connective tissue, like the blood vessels and the connective tissues, they all come from the lateral mesoderm. So if I have to produce upper limb, look at me very carefully or try to understand what are the structures we need for the upper limb. We need bones coming from the lateral mesoderm. We need muscles coming from the paraxial mesoderm or the somae or the myotome. We need nerve, and nerve is coming from ectoderm. Nerve will be coming along with so the nerve we got it, muscles we got it, bone we got it, connective tissue like blood vessels and the others, reticular tissue or whatever you know, these tissues are coming from the lateral mesoderm. So now the upper limb is complete. Similarly, lower limb will be the same thing. The bones are coming from the lateral mesoderm, muscles are coming from the myotome, the other structures like these connective tissue, etc., also from the lateral mesoderm. And how it is produced? It produces like a swelling in the two places, cervical area and the lumbar area, and then it protrudes, it enlarges from the bud to the pedal to the plate. And then it looks like a leg or the arm or whatever it may okay, upper limb. Then it rotates. Upper limb rotates laterally, the lower limb rotates medially. So the knee comes anteriorly, but the elbow goes posteriorly because it's lateral rotation. This is lateral rotation. So the elbow goes posteriorly. So that's the story about the formation. Now I have not shown any picture, so you in your mind, I understand if I were a student, Dr. Paul would like to see some photographs. Yes, I know that. I know that I'm going to show you all the photographs one by one. Make sure that you understand it. It's not difficult at all, but keep that in mind that we are talking about the paraxial mesoderm, right over here. And I try to put it like a ball, which is a somoid, which has three parts: sclerotum, myotome, and the dermatome. Now, out of three, which one you think that should stay over there, should not move? Myotome obviously is moving, dermatome is moving, the sclerotum stays over there, so the vertebrae stays over there. Otherwise, vertebrae will be over here. There's one vertebra over here, but it's moving. It is not. There's no vertebra. It stays over there in the somoid. That's the only structure stays over there. The function. Now you will enjoy the photographs one after another. It's a very simple thing. Keep in mind the story. The story is that two places it swells up. Why? That all I'm going to tell you that. And then it forms the bud, paddle, and the plate. And the inside the structures have the bone. Bone comes from the lateral mesoderm, connective tissue lateral mesoderm. There will be muscles going into that, coming from the myotome, right? The nerves will be going along with that muscle that is coming from the ectoderm. Ectoderm means nerve tissue. You know it already. So if I say radial nerve, you'll say in the exam I say the radial nerve is supporting some muscles in the extensor muscles. So the extensor muscles coming from where? Myotome. Radial nerve is coming from where? Ectoderm. They are connected together. So you need nice photographs. This photograph is just showing you that it is folding. Again, this is the paraxial mesoderm. This is intermediate mesoderm, plate mesoderm, and it is divided into two parts. It's somatic and the splanchnic. With splanchnic is forming the smooth muscle, you know it already, and it is folding along with the endoderm. So that picture is okay. Now let's see that. And you can see the somoid over there is like a ball. Isn't it? If you see the paraxial mesoderm, it's like a ball. And the lateral mesoderm is forming, lateral plate mesoderm is forming the two parts. You can somatic and splanchnic. Is that okay now? You know it already, the architecture. But what I wanted to emphasize, the somoid is like a ball. Somoid is like a ball. You need my diagram. The somoid, think about a Mercedes sign, three part. Look at the Mercedes sign. This Mercedes has a three part: sclerotum, myotome, and dermatome. Now, out of my photograph, which one should be vertebrae? Which one should be sclerotum? This one? Isn't it? Because it will be joining with this, and this will just fall down. This neural tube will fall down in between the two sclerotum to form a vertebra. Is it interesting? Whereas these two, I put it as a dot line, it will move out. It will move out as a myotome and dermatome. Is that okay? Good. So let's write it down. Somoids are bilaterally paired blocks of paraxial mesoderm. Say yes to me.
Yes.
Everybody yes?
Yes.
There are almost 40-45 somoids present in the early period, remember. They are not the final period. Early period, then it will be forming like around 30-31, presenting along the head to tail. So in that sensation, each is divided into sclerotum, myotome, and dermatome. That's exactly what I said, what I have written. Like embryology, if you should finish with me, we're in the class. You don't have to take it to read it again before the exam. It's in your mind that what is the concept behind that. The concept is that myotome is present within the paraxial mesoderm. It has a three-part, and three parts are important. The sclerotome is making the vertebrae, so it doesn't move. The other two just move out because they are, everywhere is the skeletal muscle, and the skin and the dermatome. So this is exactly what I said, and I'm reading over there. Now this is a beautiful picture. It's an excellent picture, and you can see these are the somites. So if it is joining with this one, that will be one vertebra. And see where is the spinal cord is in between. So the somites are like this, and the sclerotome is over there, and this neural tube is coming and then it's joined together. So the spinal cord is within the vertebrae. And the spinal cord, spinal within the vertebra, and a vertebra means two vertebrae joined together. The body is formed because of the two somites joined together, sclerotome joined together. And this is the beautiful picture over here. Now, if I put this photograph, remember the dissection is from the back. This is the posterior part. This is the dorsal part. Other side is the ventral, the dorsal part, the back. And take an embryo and dissect this way, then you one by one layer will be done. What is over here is the skin, neural tube, and the somite, which is the mesoderm in origin. Are you okay with that somite? Like a ball both sides. Why both sides? Because you have the intermediate mesoderm, paraxial mesoderm on the two sides. Two sides. That's what the two somites. Over here, if I put my, this will be now the intermediate mesoderm and the lateral mesoderm, which is holding ventrally. Now, I put this photograph to indicate that the bud is coming in two places, cervical area, and this is around 4th week maybe, between 4 to 5 weeks, around the end of the 4th week. Now, I tell you the story, what happened. Remember, the limb development starts at the 4th week. Now, you have to understand what do you mean by 4th week. First week is 0 to 7 days, second week is 7 to 14, third week is 14 to 21, and fourth week is 21 to 28. Limb development starts 4th week. The early part, the upper limb starts. The late part of the 4th week means around 28 days, the lower limb comes. So in this picture showing in the end of the 4th week, you agree with me? Well, I can see both the limbs, lower limb is over here, upper limb is there, lower limb is over there. This is the lateral limb. So this is the end of the. So 2 days later, this is maybe 20, 25, 26. This is 27, 28. 2 days later, the other, the bud is starting for the lower limb. Are you understanding it? You're not saying anything to me today. What's happening? What I'm saying is, the limb development, as I indicated to you, starts at the 4th week. 4th week means, don't think anywhere. 4th week means 21 days to 28 days in between. First, the upper limb comes, 25, 26 days, and 2 days later, the lower limb comes. The end of the 4th week is the lower. So this photograph is more of a 4th week, end of 4th week. Is that okay? Now, if I take a cross section, then I see everything is very obvious. This is the somite. All three things are somite. Now, outside is the dermatome. There is a different view. I've made it more simplistic way, but this is more rational to believe this is correct figure. Dermatome, myotome, and the sclerotome. The sclerotome stays over there, and this is obviously the notochord. You can see the notochord over there, the neural tube, and neural crest, all kind of thing is there over here. So it's very clear, and the picture, is cut over there. Through the somae, it is taking a cross section over there. Are you okay with this diagram? Clear? Okay. This is again in. Now you can see the sclerotome is here. The other two things, myotome and the dermatome, they start going out from the somae. They are going outside. Now when they go outside, when they start going outside of the ball somae, ball type of structure, they're going out. I'm talking about the myotome and the dermatome. They're going out, but they cannot go out completely. They have to hit which structure? Ectoderm. When they hit the ectoderm, there is a little swelling called apical ectodermal ridge, R-I-D-G-E. Ridge means swelling. And this happens at two places, cervical area and the lumbar area. That's the starting of the limb development. Limb develops when the myotome and the dermatome, they come out from the somae to hit the ectoderm and what we call as the apical ectodermal ridge. And all the genes are expressed at that place. There are many genes over there, fibroblast growth factor, WN2 gene, sonic hedgehog gene, retinoic acid. They jump onto that place and the bud starts. Bud becomes petal, petal becomes plate. I'll repeat again. And it's written over there, but I'm giving an orientation. Remember the fact that the somites are the ball structure. It has three parts, very important three parts. Sclerotome, which stays over there because vertebrae will be there. The other two parts are named as myotome and the dermatome. Myotome, dermatome go out from this. But in two places are very important for the myotome, dermatome to hit against the ectoderm, because outside is ectoderm. You can see that. What we talk about, this is ectoderm. This is ectoderm. You got it? This is ectoderm. So it hits against the two places because we need a cervical area. This is also brachial plexus come from the cervical mostly. And the lumbar plexus, so the lower limb start. The little projection. It's called apical ectodermal ridge. Very important, underline 10 times. If I say, what is the stimulus? What is the triggering factor for the limb development? Your first answer will be in the MCQ, you have seen it, apical ectodermal ridge, go for it. Why? Because the moment the little ridge means swelling is happening, then all the genes are expressed over there. The genes like sonic hedgehog gene, WN2 gene, retinoic acid, fibroblast growth factor, all these things jump onto that, and then the muscles come over there, bone is forming over there, connective tissue is forming over there, and it will look like a limb, upper limb or lower limb. This is the story. Let me write it down there. What is there? You can see now dermatome has gone quite far away from this somae. Only sclerotome is there. This has gone out, this is gone. But they together, they will be going somewhere else. The formation of the limbs, let's say, developmental limbs begin near the end of the fourth week, 25, 26 days, I said. This is the upper limb, the lower limb starts 2 days later. Homeobacter genes regulate the pattern of formation of the limb. Not important. Limb birth from the apical ectodermal ridge. This is underlying that, and is initiated by retinoic acid, fibroblast growth factor, WN2 gene, and sonic hedgehog gene. These genes jump onto the apical ectodermal ridge to form the limb birth. Main initiating factor is the apical ectodermal ridge, which is the most important part. But it is initiated by retinoic acid, fibroblast growth factor, WN2 gene. Both paraxial and lateral mesoderm participate in the formation of limb. So the limb development is from the paraxial mesoderm because of the muscles, and lateral mesoderm because of the bone and the connective tissue. All together they form the bone. So in the exam, this is a frequent question I ask, the limb development, the last line is very important. Both paraxial and lateral mesoderms participate in the formation of the limb. This you have to understand tonight. Tonight, you have to understand that what Dr. Paul is talking about is why paraxial mesoderm? Because of the myotome. Why lateral mesoderm? Somatic part especially because of the bone and the connective tissue. Both somatic and lateral plate is important. Both somatic and splanchnic both participate in the formation of the limb from the lateral mesoderm. Okay. Now, do you like this picture? This is showing you the two places where it is forming the apical ectoderm. This is not from the book. This is, I view this way, that this is the students will understand that in two places, we do not have the link from here, not from here. And the two distinct places are the two distinct places AER is activated. That's it. This is a very good photograph. That what is happening. Upper limb, lower limb. If this is the limb, this is the 25, 26 days, and this is 27, 28, end of 4th week. The limb is complete. Complete means the starting point is there. Then 5th week, 6th week, 7th, then progression will be there. And by 8 weeks, everything is complete. This is scrotum joined together. Are you getting interested in learning it, or it's just boring? It's very interesting, that's the way. And then the rest, if you understand this part, my job is finished. And I can show you more photographs with this. I have beautiful photographs. I draw by myself to make sure that the students understand this. Formation of apical ectodermal ridge, the starting point of limb development. Beautifully written. Starting point of limb development. Apical ectoderm. Often we call AER, apical ectodermal ridge. Remember this man. Remember it. The two places. And then you see the nerves are coming from the ectoderm, going to the muscle, to the myotome. This is the myotome, and this is the bone from the lateral mesoderm. And then they grow. Now it's more like a paddle, and then the plate will be there. So these are the story of the limb development. The limb development is initiated by the AER, apical ectodermal ridge, where all the genes are expressed, and then the myotome comes over here, and the bone is formed from the lateral mesoderm. And that's it. And then it will look like a bird to a paddle to the plate. And I have that photograph too. I come back to that because like my diagram, bird, paddle, plate. Plate like this. Then there will be apoptosis to form finger. Very easy. It will be originally this thing. This is after the apoptosis. 1, 2, 3, 4, 5. Yeah, I have 5 fingers. It will be there, then apoptosis. I come back to that next class. Not today. Today I'm giving the overview. Okay? So let's see what are the slides I missed it. So this photograph is clear? Say yes to me.
Yes.
Sure?
Yes.
Now let's read it one more time, okay? Formation of upper limb happens first, cervical area. Remember brachial plexus. You learn that because you cannot remember it. Usually I used to have my first lecture in the second week. This is second week, but it is really first week for you. You had only one day last week. This is the first week. I usually come after the second, so then you know what is the brachial plexus. It's a very interesting part. Will be taught to you. I think this week itself brachial plexus is there. So that comes from the cervical area, most of the cervical area, and 1T1 is there. But mostly from the C5, C6, C7, like that area, the brachial plexus. Formation of lower limb happens after 2 days. 2 days means if I say that 25, 26 days is the upper limb, then the lower limb will be 2 days later. Is the 27, 28 days. Lumbar area, remember lumbosacral plexus. 2 areas. Connective tissue bones are formed from the lateral somatic mesoderm. Say yes to me.
Yes.
Everybody understands it?
Yes.
I'm very happy, like, if you understand this part, it's a difficult part, The other things are very easy. That how the limb is developed. It starts with the paraxial mesoderm, start with the somat, understand the Mercedes sign, the 3 parts over there, and the 2 parts move out, and the sclerotum is there to form the vertebrae, and the other 2 are doing what? One is the dermatome, another the myotome, going to the limb area, or anywhere. If I see the anterior abdominal wall, is coming, all the muscles coming from where? From the myotome. From the myotome. Dermatome and myotome move from paraxial mesoderm. Yes?
Yes.
Myotome, paraxial mesoderm is responsible for all somatic and skeletal muscles. So everything is written over here. Everything I told you also. Not difficult at all. So this is exactly photograph that you make. This is just to show you that myotome migrates and forms skeletal muscles of the entire body and attaches on the bone. So this is just to show you that how the nerves are going along with the myotome. Some of the muscles, you will learn it. The nerves are, the 3 nerves are going for a muscle. One nerve, sometimes 1 nerve, sometimes 2 nerves, 3 nerves are going on. So these are myotome distribution. I'll explain that more in detail next class, not today. Today you get the holistic view of how the limb is developed. 4th week, what is happening? 4th week means 21 days to 28 days. In the middle, the upper limb, later part, lower limb, 4th week is done. End of 4th week. So if you see the upper limb and lower limb, both are there, it must be end of 4th week. Beyond. You know the limbs? Now quite a bit of written, and let me read it out. I don't love to read, but here I'm reading it. Soma is formed on the paraxial mesoderm and then begin to differentiate under the influence of genes forming sclerotum, dermatome, and myotome. The lateral portion of the myotome age migrates and level of limbs, upper limb first, then lower, and mixes with the somatic lateral mesoderm. Meanwhile, the dermatome continues to contribute cells of myotome. The apical ectodermal ridge forms after a contact of mesoderm and ectoderm and initiates limb development through the expression of various genes. Basically what I said is there, that at the end of the day what you want is the 2 bud initiated by apical ectodermal ridge. That is, the ridge means swelling. That swelling, when it is there, it is under the influence of genetic expression, and all the genes come over there, like sonic hedgehog gene, retinoic acid, fibroblast growth factor, WNT gene. All these genes are there. And what they do, they will be increase the length of the limb. We call the paddle shape. So paddle shape means there should be a bone coming. This is also under the influence of the gene expression. And the bone is coming from the lateral mesoderm, including the connective tissue. So that's why it is written over there. Limb development occurs at different times for forelimbs and the hind limb. In the mid-4th week upper limb, so 25 to 26 is the middle of the 4th week. First, form and lower limbs after 2 days. The limbs form at vertebral segment where C5 to C8 is the upper limb and L3 to L5 lower limb. You learn that later in the 3 stages of limb development on apical ectodermal ridge: paddle stage, grade stage, rotation stage. This is, I just said that, I forgot to write BART. You can add over here, okay? bar, paddle, play, and the rotation. I forgot to write that. You write it. First is the swelling little bit, bar, then is the paddle, and then is the rotation. Remember the upper limb rotates laterally so that the elbow comes posteriorly. The lower limb rotates medially. So the baby was like this way, then it was like this way. So then the knee is coming anteriorly. Knee means knee joint. And this is exactly what I have to put up there. Molecular development, limb initiation, apical ectodermal ring, fibrous growth factor. In detail, you don't have to know detailing, but remember there are multiple genes are responsible for that. You are also the influence of sonic agent gene and the expression of maintaining the axis of the limb. These many books have written very detailing of this. We do not now, students do not have to know detailing of that, so long you know multiple genes are responsible for the development of the limb, but it is the initiation factor is apical ectodermal ridge. And then all the genes, if you remember, is good, if you forget, is better. You don't need so many genes. But appreciate the fact that there are multiple genes, sonic agent genes, you know it already, WN2 genes, and then you have fibrous growth factor, retinoic acid, you know all these genes. It's not an alien name. You know everything in detail earlier. The upper limb rotates laterally through 90 degrees, so the future elbow point dorsally. Dorsally means backside. The lower limbs rotate medially through 90 degrees, thus the future knees face ventrally. Are you okay? It's very interesting. I don't think I should go further. Yeah, beautiful. Is that okay? I think because it's a beautiful picture, I stop it over here. Thank you very much for listening to me.